Yeah, because there's no one.
Last the ones that are coming, I don't watch.
Yeah.
Hey, Prabin.
Hi, Mitch, everyone. My audio coming through okay?
It is.
We're going to do a quick test of the slides as well.
Perfect.
All right, give me a second.
Good, how are you doing? Hey. All right, pretty
It's good, Prabin.
The full slide is up, right?
Yeah.
Yeah.
That's
Yeah, we spot. In town,
everyone's talking like.
yeah. Better come to the lab meeting.
How's it going? It's going. Officially start your job? Officially. Been sound out the last meeting. Oh. So. Oh, you got your. And then you changed your, you got back here and you're like, oh, early is October finally, and I'm gonna drive back. Forget this business.
Yeah. Alrighty, it's noon. Let's get started with some updates. I don't have a whole lot to share, although across the hall, the Misa space is being drastically remodeled. New furniture is going in. That's going to be a shared space for Misa, basically the whole Misa umbrella. So Misa proper, Greenlands Blue Waters, Forever Green, and it's going to be set up with, you know, like a conference table and kind of hoteling spots for people to come work. So, you know, mostly the core folks that I've mentioned, but if you're looking for a new place to sit for a while, change scenery, hang out with some people, come on by once it gets going in the next week, I guess. Hopefully it'll be pretty much done today, and then we'll start moving in basically. So, so yeah, it's exciting. Anything else to share? Any other updates?
I'm going to introduce Craig C., a new postdoc in the lab, working on Forever Green topics. Craig, do you want to tell people what your background?
Sure, yeah. So I guess my background, I actually did my PhD next door with Sarah Hobby and Peter Kennedy. So my background is kind of mycorrhizal fungi, ecosystem function. This is my first venture into agriculture, so I'm gonna be writing a lot of notes on terminology. I think. Yeah.
Forever green stuff.
Yeah, all of it.
Can you give us a hint?
Yeah.
The answer to that is no.
Yeah, the surprise of becoming no.
Converting to acres is the thing, yeah. This is what I was like throwing out.
And
Things, you'll get there. I got there.
there's another new postdoc in the lab, Hannah Rush. She's going to be late, but she will be here. I was going to introduce her to, has some history at the University of Minnesota, but that was in Florida for 10 years, finishing her PhD, and now it's back for a postdoc position, working with Jeff Oishel and I on some alfalfa projects, funded through ARS.
Cool. What was your name again? Remind us.
Oh, sorry, Craig C.
Craig C. Craig. Welcome, Craig. Any other updates?
So Jeff Berg here from Minnesota Department of Agriculture. So just to let you guys know, you may be tracking this already, but we had a request for proposal for clean water crops marketing, which was passed legislative session. You know, the period just closed. Can't talk about details, but we should have a nice group of competitive applications to further market development of forever green crops. So thanks.
Thank you. Fantastic. on that note, the Forever Green RFP proposals are due October 30th. I've seen lots of chatter and conversations about groups coming together to submit, so that's promising.
Can I just, I'll email you. I have one like very detailed question about the RFP.
Okay,
If that would be relevant, I can ask now if it would be relevant.
well,
but
do you think it has broad relevance? You can talk about it now, or is it too,
It's kind of a niggy like word count thing.
oh,
Is it? So there are a lot of, for anyone who isn't working on these, there's a lot of supplemental documents and there's a requirement for a diversity statement. Is that part of the 4-page word limit or not? Because it used to be.
no, it's not. Yeah, that's all in the supplemental info now.
That's all, so yeah, that's why I was confused, because it was in the supplemental information.
Yeah.
So it's not, so anything in the, so any, is that a blanket, anything in the supplemental information? It's as if I was not part of the proposal preparation.
Right.
Maybe things I should have asked a month ago.
Yeah.
So anything that supplements is not part of the 4-page limit or the 2-page limit, depending on which grant you're writing.
Correct, yeah, good clarification. That's
That's
one
all.
example of there are a number of new structural changes to the RFPs, but you can't just copy and paste an old one. Just a word to the wise.
Yeah, I did not do that. which
Did
I-
you give an update on the event on Monday?
Can I remember Monday?
The internet is next coming.
Well, do you mean last Monday or the upcoming Monday?
Upcoming Monday.
Yeah, well, upcoming Monday morning, interim president Jeff Edinger will be coming to visit our program. Along with a couple, 2 or 3 different U of M VPs and high up people who work with them, director of government affairs, and outside folks from Greater MSP, which is like a regional economic development group. So Peter Frosch, their CEO, will be here, and 2 other people from Greater MSP. They're helping me down. They're visiting us, Plant Protein Innovation Center, the microbial manufacturing facility,
And a nanotechnology center on Minneapolis.
and I think the MRI center too, I think was on the list. So five places. We're one of 5 centers, initiatives, efforts being highlighted as a key point of connection with the regional economy and innovation and science. So it's very cool. And Peter Frosch with Greater MSP, they launched recently a sustainable aviation fuel hub to boost production of SAF in Minnesota, basically. So we'll be highlighting our winter oil seeds because those are a great potential feedstock for sustainable aviation fuel, but also telling the overview story of what Forever Green is. This past Monday, something to share, the Forever Green commercialization team plus me, so Sienna, Catherine, Andrew, Matt, and Colin, as well as the hazelnut team, Lois, Mark, and Les, we're down at Kurt Kimber's farm in Farmington, so less than an hour away. And it was pretty magical. We planted 400 hazelnut seedlings. I mean, the work was hard, but it was magical. We planted 400 hazelnut seedlings. That's preparing the ground for next year planting clonal material from our top selections. And it's the Kimber Contours farm. It's an organic farm. It has contours, as the name suggests. This hazelnut planting is going into an old Kernza field that will be alley cropped until the hazelnuts take off. And it's between 2 prairie strips. So there's 20-foot wide strips of prairie plants upslope and downslope from this planting. So when that all gets going and is established, it's going to be, I mean, a leading location in the state and the region for regenerative agriculture in my mind. All the pieces that are coming together there.
And you'll learn more about this concept called Go First Farms in terms of the leading farms and the development of the hazelnut industry that moves in our new germplasm and moves them on to commercialization. So it's going to be called the Go First Farms.
Which is not, despite how it sounds, a play on gophers. The term "go first" was made up by Jason Fischbach, who's at UW, but everybody who hears it thinks, "Oh, gophers." No, it's "go first." Like these are the farms that are going first.
farms.
But he's also a graduate of
I
this
know.
program, so he probably it just happened.
Some subterfuge.
Like subliminal
Yeah.
thinking about rodents.
All right, anything else before we turn it over to Prabin? All right, take it away, Prabin.
All right, welcome everyone. We have a lot of things to cover, so I'm gonna get started. I typically present early, like January, February, so I don't have a whole lot of data. We're still in the process of threshing, cleaning our stuff, but this should still be a good update and good overview of what's happening in the Kernza world and some other perennial cereal crops. Just a rough outline. We'll focus a lot on Kernza, starting with breeding and variety trials update. Then I'll talk about Spark Project, which is a very cool name, thanks to Jake Younger's group. And then some recent, very recent mutagenesis work that we've just begun and quite interesting developments there. Then we'll move on to cereal rye, oats, and wheat, which are all perennials. And some work on some species are a little further advanced. On some, it's just beginning, but I think it's exciting nonetheless in all of these. A brief introduction to intermediate wheatgrass. The species name is Thynoparum intermedium. It's a perennial, very, very distant relative of wheat. It is immune to a lot of the rusts that comes from small grains, but it does get infected by scab and other blotches, leaf and stem diseases. It has been used to give or transfer genes from the plant to wheat, and it has excellent winter hardiness, which is one of the reasons why we're working on this crop here. There's also a breeding program up in Canada, and they also see just about the same level of winter hardiness that we do here. It is an allohexaploid, just like wheat, with 3 parents and 3 subgenomes. The genome is sequenced. We now have a much better, much more refined genome sequence. It's still and we're still calling it draft, not published yet, but thanks to collaborators at the Land Institute, Kansas State University, Hudson Alpha, we have a working draft that we use for our genomic analysis. It's a fairly large genome at 12 gig base pairs. Compare that with wheat, which has 16, so almost up there. It is a near obligate outcrosser, and because of that, most of the population is quite heterogeneous, with almost 35 to 40% of the genome being heterozygous. And the panels, those grasses on the right, they are the 3 progenitor species of intermediate wheatgrass. Here's a quick timeline of domestication. It was brought to North America as a forage crop, and the domestication process didn't begin until the mid-80s, when Rodale Institute started some work, and then later on in early 2000s, the Land Institute picked up the work, and we started our work in 2011 from the materials Land Institute provided us, and since then we've completed 5.5 breeding cycles. We're in the middle of 6th. The 7th is ongoing, and in this time frame, we've released 1 cultivar, and another one is going to be released fairly soon. End of this year, it'll be proposed to the crop variety review committee, and then hopefully, if there's approval, it'll be our next variety. And I'll talk more on that a little later. These are the breeding goals for Kernza, but I'd like to highlight not just for Kernza, other perennial cereals. The target is about the same, where we want high grain yield, especially in Kernza. We want a sustained or persistent grain yield, because many of you probably know in the 3rd year of Kernza cultivation, the yield goes down quite dramatically in our climate, and it's a challenge we're trying to tackle. So that's important. Of course, important is seed size, resistance to shatter, as we do not want to lose the grain in the field. And then free threshing, which is the ability of the grain to come out. It's basically dehulled grain. We want that as high to be possible. We're also keeping an eye on spike traits, spike length, spike weight, as they correlate positively and significantly with grain yield. Plant height, people talk about dwarf plants, dwarf wheat. It seems in Kernza there's a negative relationship between height and yield. So we want height to be optimal, but that's not to say in the future, there's not going to be a Kernza plant where short plants aren't going to yield more, but that's probably a long-term plan. Up here, we also look for lodging resistance, as we do have we get decent amount of rain in our soil. There's good moisture. I keep an eye on diseases, mainly scab, also ergot. Anything, it's mostly a presence absence kind of data. With if there's disease present, then those are tossed out. End use quality, analysis of nutritional components, storage ability, and what food products can be made. All those work are done in Department of Food Science, mainly by Pam Ismail and George Anor. And I think they'll be giving their own individual presentations as the semester progresses. So some breeding updates on Kernza. We are in the middle of evaluating cycle 6. As I mentioned, it has about 1,000 genets or genotypes in a spaced plant fashion, as shown in the picture. The top right is St. Paul and bottom is Lamberton. We have we collected data this summer. We have harvested materials, and those we're measuring and cleaning and threshing and getting yield and seed size information on that. The text shows what traits are measured at what point. I'd like to go through those quite quickly here. Spring growth vigor is early May. By 10th of May or so, it's taken. Heading as they emerge, which is early June, and by mid-June, they're almost done heading here. Spike length, number of spikelets, those are also done in the field. Spike length, once the head is fully, number of spikelets once the head fully emerges. But spike length, we wait maybe a couple of weeks before harvesting so that they're done growing. Plant height around the same time as well. Then plant health, scab, ergot, I go through the field as I see them and then take notes. And then most of the other work happens post harvest and post thresh, including shatter resistance, spike weight, free threshing. Ergot is also done during threshing. When they thresh, you can see the ergot kernels with the sclerotia quite clearly. So it's easier to take notes on that at that time. And then of course, the yield, seed dimensions, those are also done after threshing. Cycle 7 is underway. We planted about 1,200, so 200 more than cycle 6. And compared to cycle 5, this is almost twice increase in size. We had about, I think, 576 individuals in cycle 5, and now we have easily doubled it. And the goal is that a larger population means chances to see better genotypes, the genotypes that have the trait combinations we are after. And every year from next year onwards, well, actually from this year onwards, it's going to be one cycle a year, about the same population size. What we're doing also different, starting with cycle 6, so the previous cycle, is that we have the parents of the breeding populations also present in the field. And we didn't do that in the past, don't know why, but we're doing it now because it's shown in most other crops that when the parents are included with the progeny or the breeding population in genomic prediction models, the models perform better, basically helping us make better selections. So they're evaluated together now. So phenotyping of cycle 7 will be done next 2 years, and so on. So you can see the 2 plots in Lamberton and St. Paul. The variety candidate performance data is from last year. As I said, we have yet to collect data on these. We're currently in the process of threshing and taking yield and seed size measurements. But to go through this briefly again, the yield data shown here is relative to MN Clearwater, which is shown in green right here. The best performer across 2 years in Lamberton, Roseau, and St. Paul in 2021 and 2022 was MN 1607, but that candidate had some poor stand issues. I think the data is a little flawed. We planted a large increased plot, and it didn't do too well, so that was discontinued quite quickly. Also has smaller seed size compared to others, and I'll talk about that in the next slide. The next, right next to it is MN1603, which is the candidate for release here. And across 2 years, it's roughly 6 to 7% more yield than MN Clearwater. In year 1 alone, it's about 13% more yield. So as you know, you work on perennials, you have to look at multi-year performance, and it looks that in 2 years we have about 6 to 7% more yield. Seed size, this is also 2-year average. A roughly 6% increase in seed size on 1603 compared to MN Clearwater. If you look at just the first year performance, that's about 11%, if I remember right, larger seed size compared to MN Clearwater. So year 1 is always the best performance, so to speak, and then after that it dips down, with year 3 being pretty bad, and it stagnates after that in terms of how worse it performs. And 1607, the one that did the best in terms of yield, it has, you know, it's respectable, but smaller seed size. I should add here that 1603 currently is being increased in large acreage, 5 acres each at two places. One with Carmen Fernholz, who is an organic farmer in Madison, Minnesota, about 3 hours west of here. And another site is Rosemount in coordination with MCIA. We're hoping to get enough seed next summer for another round of increase and or release to the growers directly on a smaller scale. We have to go through that discussion yet, but we are trying to increase the seed at multiple sites. We also have new variety trials underway. I myself have 2 locations, and Dr. Jet Youngers has 3 locations with new candidates. Some of the materials have also been sent to Lee DeHaan at the Land Institute, and we'll also be evaluating our new candidates. And new trials should be established next year. I've talked to Don Velikson to put some new trials up in Drozo. I have yet to give him the new seed. So it'll be a very broad assessment of the new candidates, MN19s, MN22s, and the ones after that. Want to move on to a SPARK project. It's SPARK stands for Space Plants and Rhizotron in Kernza. These cool names come from Jake Younger's lab. They're really good at giving really nice names. And I would have called just mini Rhizotron in Kernza, but you know, I'm glad I'm collaborating with Jake's group. The main goal here is that we want to study the roots in space plants in our breeding population. One, to know what's going on. Is there any changes due to breeding? And second, of course, is to maintain wheatgrass's potential for environmental services. As we select for above ground traits, we do not want to lose the below ground traits. So we just want to get an assessment of what is happening. So we installed 160 Rhizotron tubes in October 2022. And imaging began earlier this year. The main idea is to have 3 sets of images, one early in the spring that was done in May, a second one right around harvest that was also done, and then the third one is in the fall, right after the freeze, so that there's a sharp reduction in root activities and get a snapshot of what is happening at that time. So the third imaging is yet to be done, of course. It'll be done in early November, and then we'll repeat the whole work next year and use some machine learning methods. Jake's group has already developed a fine-tuned a method where the images are brought in manually. You have to curate some of the images, and then the machine learning algorithm takes over, and then it extracts information from the images, which is then fed to yet another software to then grab like these parameters such as root number and length and diameters, area densities, and so on. And then we will put the root data along with above ground data and see what kind of relationship we observe. I'm expecting there will be a segregation or variation in terms of how the root traits are, given that Kernza field. If you've been there, each plant looks very different than another. We have all sorts of plant combination in this field, from short dwarf types to tall, bushy, tillery, non-tillery, and so on. So there has to be some sort of differences, but to what extent, I'm very curious to find that out. And the training of the machine learning models with images taken this year will begin, I think, as early as mid to late October. The bottom picture on the slide is also another very intuitive way of labeling the plants that have been sampled. So very cool. A mutagenesis work. This is a brand new work that David Marks is helping us with. He treated dehulled grain of MN1605. This is not the candidate to be released, but it was a second in the list in terms of performance. It also has as large C as 1603. So he treated naked grain with EMS in 3 dosages, so 0.1%, 0.2, and 0.3, and was treated for 18 hours at room temperature and mixed under chemical hood. You can see the picture on the right there. And what David did was he planted some seed on trays or flats. So there's 2 replications in each tray there. Each tray here there's roughly 20 seeds. If you go top to bottom, there's 20. And then if you go left to right or right to left, there's 15. So roughly 300 seeds in this tiny little guy. So about 600 seeds approximately. It's not exactly that, but roughly that number. And that was done for 3 all 3 dosages. And there's plants growing, as you can see. In the very back is 0.3, the middle is 0.2, and then the front is 0.1. And we took measurements on how many are growing. One, to assess that, is it even working, and is it working as expected? And it seems so. The treatment with the highest concentration of EMS has the least percentage of germination, so about 14% germination rate in the 0.3% treatment. Surprisingly, 0.2 has more than 0.1, but I was telling David this morning that I see that some seeds are still germinating in the trays that were treated with 0.1 and 0.2 percentage. So I think these numbers will change a little bit next week. But good thing is that there's some growth, and my plan is, and someone can tell me if maybe that's not a good plan, is to take most of these plants to the field. It's still not too cold. They should establish okay, and leave some in the greenhouse as backups. And speaking of field, we also planted some in the field in E6. That's I call the fusarium shed, but that's barberry fields here, and then that's the shed. So if you go to E6, you'll see these long plots. There's one plot with 0.3% treatment, 0.2 and 0.1 as labeled. And I took a rough measurement. I did not have a tape with me, but in the 0.3% treatment plots, it seems there's one plant maybe every 2 feet. If they're done germinating, that is, maybe there'll be more germination. You can see right here, this seedling, excuse me, and then here, and there's one guy right here. In the 0.2 and 0.1% plots, it's a little higher, maybe one plant every foot, foot and a half. So it kind of matches what we were seeing in those trays with more germination in the less dosage treatments. But overall, it looks like the EMS treatment has worked, and there is germination. And I'm curious to find out how these plants behave. I was telling David, if we can find a plant that sells with good seed set, that would solve a lot of issues in Kernza, but we will see what kind of plants grow. Before I move on, I know that David is on the call. David, do you want to add anything to the work, or what you've observed, anything based on your experience from Pennycrest mutagenesis?
No, just simply that it was kind of a fun little thing to get involved in, and I'm, happy to help out any way I can. I think the next stage will be to see if we can see any kinds of dominant sorts of changes. To get to the recessive things with the fact that it's an outcrosser, we'll need to take seeds off of individual spikes and plant those sieves together and try to get them to cross with each other and see if we can pick up some recessive mutations that way. But surprisingly, I think I probably could have gone higher on the EMS. Maybe next time we'll do that.
Yeah, the very first time we've done this at the University of Minnesota, so we don't really know what to expect. Yeah, maybe next
For
time
sure.
higher dose or
Yeah.
and going back to the point of what you said, many of you probably know this. Kernza is largely predominantly self incompatible, and I will be bagging some of these individual plants. And if some do produce more seeds than what I've normally seen in other kinds of plants, I think that will be very exciting find.
Yeah, I would say that what we'll need to look at actually it's likely that any individual stem has a different mutational makeup. So we'll probably have to, kind of treat each little stem that comes off the plant separately. And so that will be one thing to be aware of.
Definitely. If you see other seedlings there, that's spring wheat. I mixed spring wheat to get a sparse planting density for Kernza. So it was basically an attempt to get thinner stands because once we get frost a couple times, this spring wheat should die. Moving on and wrapping up Kernza breeding.
Carmen has a question.
Yes.
In the chat, what is EMS treatment?
Oh, sorry, Carmen, I was not. EMS stands for, I think, ethyl methensulfonate or something. David, please correct me. I do not know the full form, but it's basically a chemical that disrupts the DNA, a single mutation, random in the genome. So it introduces variation. And the idea is if some of the genes controlling, say, seed size, yield, plant height are disrupted, what kind of phenotype we can see in the field. That's the goal here. So it's not GMOs. We're not introducing any gene from anywhere else. It's just a mutant using chemicals randomly in the plant.
Thanks, I appreciate that.
So the next steps for Kernza breeding program are that we are continuing data collection in cycle 6, which we've completed 1 year. Year 2 is left. It looks like I missed cycle 7, but cycle 7 continues as well. We're crossing cycle 8 parents in the greenhouse this winter. They are currently in the growth chamber, and then early spring will be genomic selection, and then we'll pick candidates for cycle 8 population that will be planted next fall, and also the parents of cycle 9, which will be the year after. So that's sort of immediate next steps. In the summer, of course, there will be field evaluation, looking at mutants, establishing new crossing blocks. On this, we have been establishing crossing blocks in the fall, but I've determined that it's a little too late in the fall sometimes, where the clones that are disrupted from the mother plants don't get enough time to establish properly in the field, and we sometimes lose the whole next year as they don't grow as vigorously. So starting 2024, we'll be putting in crossing blocks in the spring, early summer. That way they have the whole summer to establish, and we'll get good growth the following year. So that should help in terms of variety development timeline. And then fall 2024 is establishment of cycle 8, new variety trials, and also establish increased plots for some of the best MN19 candidates, which are not the latest cycle, but a cycle before the latest cycle, as we'll have 2 years yield data by that time, hopefully. Okay, moving on with perennial cereal rye. It is also a novel cereal crop, entirely human-made, as it was crossed. It's a cross between Sacchalia cereale, the regular winter rye, with Sacchalia montanum, a perennial type. in the 60s in Germany, and they did a bunch of backcrossing testing and evaluations for perenniality, large seed, less shatter, which shatter is still a problem actually, more so in this than in Kernza, funny enough, and for yield. So grain yield, seed set, grain size, those look pretty respectable for a crop that's only about 4 or 5 decades, but also the fact that Sacchalia cereale was already domesticated, that also helps quite a bit. It has dual use potential, similar to winter rye, all the traits of winter rye, plus the perenniality, if you want to imagine it that way. That said, at the bottom, I say it could offer ecosystem services because we do not know yet. We have not done any of these tests. I plan on working with Jake Younger's team and trying to evaluate some of these things. We don't even know agronomically when to plant, how much depth and so on. But again, the fact that one of the parents and most of the genome coming from Sacchalia cereale, it should be quite similar to winter rye in how it performs and behaves. As I mentioned, it's more similar to Sacchalia cereale for the genome. There's diploids and tetraploids, so both types of populations, quite stable. They both exist. I have not crossed them to each other. I don't know how well that works. And it's also an outcrosser. But from my experience in rye breeding, very brief one, it can self up to 3 or 4 generations. Is that the same case in perennial cereal rye? We don't know. I'm starting some selfing in the greenhouse this year, so I think we'll find out in as early as maybe next fall. There's some pictures from our evaluations in the past. There's quite a bit of diversity in these plants or in the population as well. These are both diploids, and you can clearly see the height difference, the biomass difference. There's diseases and lodging and all sorts of things you can imagine. There's ergot. It's again, any problems that we've seen in winter rye, I think, is a problem in this crop as well. So definitely needs to go through a selection process to make sure we're selecting elite plants each breeding cycle. So to summarize the work so far, in 2020, we planted about 620 plants, some diploids, some tetraploids. Those both came from Canada. There was a USDA equivalent of Canadian federal agency, and I received some seed from them. I also received about 121 Sacchalia strictum accessions from the USDA core collection. Couldn't find Montanum at that time. And I'm calling that population cycle 0. We evaluated for a couple of years, selected some individuals. But I noticed in the field that there was some segregation for perenniality. I was told that the Canadian populations would be all perennials, but not quite. So I thought, first, let's make sure the plants we have were actually 100% perennials. So from the bulk harvest of cycle 0, planted about 2,200 seedlings. And after 2 rounds of cold chamber, so essentially exposing to 2 rounds of winters, about 850 have survived, and all of them are in the field, the same E6 field that I showed for Kernza. And I'm calling this population cycle 1 officially, as they should be at least 2-year perennials after going through 2 rounds of fertilization. We will look at the population next year and the year after, with some candidates also selected to establish maybe small crossing blocks, just to see how does that process work out. Do we need to follow, can we follow a similar model we have for Kernza, or do we need a separate model for population improvement and variety development? And at the bottom, you can see that the nursery design, so far, it mimics wheatgrass, but I want to move a bit more away from the field in the beginning, do as much selfing and crossing in the greenhouse, get up to F3, F4, F5, depending on how much selfing and crossing I can do, and then move on to row-based phenotyping in the field, as I did as when I was working with rye in Germany for a while. Yet to be determined. but at least in next summer, we'll evaluate the population that we planted this fall for important traits. And again, I listed the number of traits for Kernza. Most of those will be similar, except free threshing. This one threshes really well, just like winter rye. And we'll establish some crossing blocks, and then by selecting the best performers next summer, and bring them to the greenhouse, and then self as well as initiate by parental crosses, maybe a small group open pollination to mimic this synthetic crossing. Again, that's to be determined. I'm just trying different things and see what is the best approach to take rye towards. And the long-term plan, fall 2025, you know, there's some S2s and F2s, but it'll be basically generation advance selection, and back crossing and that kind of work. Okay, Don probably has been rubbing his hands when he sees this. He's been the most excited about perennial oat. The work on perennial oat is it's in a very much baby steps. We haven't done a whole lot, but I think pretty exciting. It's the species of Avena macrostacea, and from what I can gather, it's the only perennial and outcrossing species in the entire Avena genus. Maybe there's other species that have yet to be discovered that also do the same, but so far this seems to be the only candidate that does both things. You can see the spikes on panel A. It's from paper. The citation is at the bottom. The plant stands in the middle, and then the panel on the right shows the seeds. Very, very tiny, tiny grain, and we'll look at more pictures soon here. So it was discovered in the mid 1800s, and it was named macrostacea, but slightly differently. I'm just giving a history because this was kind of I was curious to learn about the species, as not much exists out there, surprisingly. And then in 1855, they renamed it to macrostacea. And 1855 is when Minnesota was not even a state yet, and Lincoln was not a president yet. So, but it has less to do with the discovery of the species and more to do with this picture on the right. I obtained this article. I think it's written in French. I'm just shocked that I can still find an article from the mid 1800s to this day. That's why I wanted to give reference on Minnesota was not even a state, but we have a paper in the digital archive that we can access. That's really cool. Anyway, so the seed is about a third. I need to update that. It's probably a fifth, if not even smaller, of regular oats. Now that I've seen actual real macrostacea, my own eyes, and a lot of characteristics are mostly unknown. We really know nothing about this crop. It's native to Algerian mountains. But I'm butchering the name probably, but the Dura mountain range or something, and some parts of Morocco, but mostly Algeria. And there's not a whole lot of scientists working on this species. If you remember, most of this information is from last year. We planted about 240 plants of macrostacea, or so-called macrostacea, that came from a farmer in Indiana. And things are looking good. They were even booting and producing these inflorescence, which was surprising. But then this was in the fall. Come next year, they all died. I don't have a picture of that, but turns out that was not the real perennial oats. So then that winter, I started searching, and Helene Murray, the MISA director, she put me in contact with a scientist in Algeria, and we've been communicating since, Dr. Sabah Razi, and we're trying to get seed from Algeria to here. She's gone on a few collection trips. So she works at University of Biskra, which is, from what I can tell, a desert city, more or less. So she's been going on these trips to the mountains to look for plants. She's mobilized her contacts, talking to farmers to see if we can find anything. And the whole summer, every other day or so, she would send me 10, 20 pictures, and we would say, hey, it looks like macrostacea. It does not look like macrostacea. Let's collect it anyway. So at some point, I'm expecting a fairly large shipment from Algeria, and some of the pictures that she took. Here's additional pictures. They look very similar to what's been reported in the literature, and some random pictures I found. Now, are they truly macrostaceous? Neither Dr. Razi knows, nor do I, until they're here and we plant them, evaluate for perenniality. Maybe if there's markers, which I doubt there are, but just to see how it compares with other species in the Vina genus, and then make some judgments from there. But they look similar. They look similar to what I've seen. And again, that's 1 centimeter here, and just look at the seed size. It's super small, and I don't see this species being a crop per se, but I see the advantage being crossed with regular oats and bring in the perenniality of this species, bring in other important genes that it could have. We don't know to regular oats. I think that's where we should focus on. Now, from Algeria, let's talk about Canada. Some seeds were right here in our backyard. Didn't even know after extensive search. I just stumbled on this global grin, and there's just one hit of all the countries in the world. Canada has the seed, and look at the collection history. The seeds were indeed collected from Algeria. This mountain range, and this was done in the 70s. There's a couple of papers published. Apparently, the Canadian scientists flew to Algeria, found these clumps, dug them out of the ground, and then shipped to Canada, of which I think about half survived. And that's how they maintain the plants, and that's how they've preserved the seeds. I asked for some seed. I got exactly 20 seeds. They're right here, and they're really small. I dared not even open the husks. I didn't want to bother them. So Brett Heim, technician working in our group, he talked to MCIA folks, and they directed him to a protocol in germinating oat seeds. Again, we didn't want to take any risk in just putting them in the soil and expecting something. So we treated with disinfectant, some acids, and so on. And at the end of the day, we were able to successfully germinate 10 out of 20. So 50% germination rate. I'll take it. I was hoping I'd get 2, but 10 is good. I don't have the 8th, the 9th, and 10th plants here, but the plants with those 1 green leaves, now they're doing much better. This is from about 3 weeks ago. They're doing very well in the greenhouse, and I'm glad that we have live plants. So the next steps then is are these plants going to be as bushy as Kernza? I don't think so. Will they get bushy enough to be cloned at least once? Instead of 1 plant, can I turn it into 2 plants or 3 plants? We don't know yet. If we can't, we'll just cross without cloning with regular oats. And I've talked to Dr. Kevin Smith, and we've agreed once the time is right, we'll get some training on oat demasculation and pollination. He will give us some seeds so we can do some staggered planting in our greenhouse. So we have plants growing all the time, and they're ready when macrostaceae plants flower. Do they even cross? I don't know. That's another thing to be tested. I hope they do. If Kernza Thinopyrum can cross with wheat, triticum, I think same Avena species. I'm hoping there's a higher chance of pollination and something happens. And then if we get progeny, if we get good amount of seed, then we can start talking about field, or growth chamber. But let's see what happens. I'll keep you all posted.
But there's evidence already, isn't there, that these crosses have been made between the 2 species with annual and perennial, that there are in fact hybrids available in Europe?
On oats, I
Yeah.
I haven't seen it. I mean, I'm sure it exists. The species have been around for a while, so I'd be surprised if someone didn't do anything with them. I personally have not seen them.
The material that came to Minnesota earlier on was from Europe and they were hybrids. Right, so I think there is a seed bank in Europe where those hybrids would be would reside, and also there's materials I think in Israel in their seed bank.
Yeah, it's all likely I'm not too familiar with this species as I am with others, but seems the work does go back to the 70s, possibly even before that if it was documented.
Hey, Don.
Go.
Yeah. I've been approached by some people in Europe, I think, that claim to have perennial oat, but it was not an Avena genus. There's things that are, it's just like, you know, ryegrass, there's oat grass, different genus, things that people call perennial oat, which were not actually oats. So I'm not, there's a lot of confusion about that. But back in the day, Stan Cox knew a USDA geneticist, I think, who had some plants and maybe had made some hybrids with embryo rescue. I'm pretty sure it takes embryo rescue.
Any other thoughts on this before we talk perennial wheat? Okay. So the last species is perennial wheat. This is mostly just evaluation and assessment of what perennial wheat does in Minnesota, and turns out it does pretty well. It's a hybridized form of wheatgrass, Kernza, with common wheat and durum wheat. I think there's some specific durum wheat type that the Land Institute has been crossing to, Dr. Xu and Wang. In 2020, Jake Younger established some observation plots north of the Horse Center, and this summer was third summer, yeah. And the plots are still in the ground, so next year will be fourth year, and they look pretty good. They look pretty good. We also had, or Jake had some materials also from Kevin Murphy in Washington State, but by and large, majority of the materials are from the Land Institute. Some images from the field. If you've seen Kernza and if you've seen wheat, this is kind of in the center. And this is one of the better spikes. There's some really bad ones. There's some better ones. But I just wanted to highlight some similarities of Kernza, which is longer spikes, and the spikelets, the arrangement of Kernza. But then you have, you know, those very obvious arrangement of wheat. Look quite well in the field. Even after the third year, 3 years of growing, the plants stayed true to where they were planted. Or in other words, you know, Kernza gets bushier as the years go by, very competitive within itself, and one of the reasons why I think we have the yield decline, but perennial wheat does not seem to have that issue despite Kernza being one of the parents. So that's excellent. Also, since they've grown for 3 years, it does really well in our cold climates, and something we should definitely consider further evaluating. So given that, we thought, well, let's put them in some sort of breeding or crossing block type program and see what we can do with it. And that's what I'm sharing here. I think I already talked about the first 2 points where the materials is obtained from. So this summer I selected about 10 spikes or so from 24 best looking plots out of 40 some plots. There's still some segregation in the plots. They're supposed to be mostly inverts, and I think they are, but you can see some have on, some do not. There's differences in height. There's definitely differences across the plots in how they fare against different diseases. So I picked the 24 best ones. We threshed, mixed with spring wheat again, and planted in these long rows in the same Kernza field. And I'll show some pictures later. But one thing I want to talk about is that despite threshing about 10 or so spikes, the amount of seeds I obtained is less than optimal. Now for a new crop, this is not too bad, but there's some selections and some improvements that need to happen. It's possible these are all selfing, by the way, so it's possible that some of the self-incompatibility of intermediate wheatgrass has made it into this this new crop. Nonetheless, exciting to see larger seeds than Kernza. Here's a comparison of seed size. And let me see. So the first, the 5 columns on the left are different perennial wheat seeds. And after that, let me try and get the laser here. This one right here is a spring wheat, Lang MN. And then of course, this is Kernza on the very right. The seed sort of, you can see where it gets the seed shape from. The length kind of comes from Kernza. And then there's some width to it, but not as much as spring wheat. So again, a much more respectable compared to Kernza, but nowhere near enough as spring wheat. And as a new crop with not a whole lot of work done, I think this is already quite good, but with selections and more intensive work, they could get up there. Here are the plots. There's 3 plots, so one here, and one there, and then there's 8 of those, so 24 total plots, 30 feet long. So if anyone wants to stop by, see how they're doing next summer, feel free. And not all of this is perennial wheat, by the way. These are also mixed with spring wheat, as I mentioned. After the cold kills spring wheat, then we should only have perennial wheat plants and also low density, so I can tell individual plants apart, hopefully. And then take the good looking ones to the greenhouse and then make crosses. And speaking of that, I had a meeting with Dr. Shu and Wang a couple of weeks ago. He's also sending new materials soon, and these I will start right away in the greenhouse and make some crosses. And if I can get F2s by next fall, like thinking about planting F2s directly in the field, so they could be evaluated in 2025 summer. I'm also going to try some crosses between the wheatgrass and wheat. I tried last winter and it was good success, but none of them germinated. But now that we have this oat germination protocol in place, maybe I'll try that and see if I can germinate some of the crosses between wheatgrass and wheat. And yeah, basically crossing and evaluating for a couple of seasons and see what we can do with it. If there's enough grain, I would also love to work with food scientists and see what kind of nutritional component or composition, excuse me, these grain have compared to Kernza and common wheat, and try to find their place in the market. Okay, with that, I would like to end here. Thanks to a lot of people across different departments, different institutions. Specifically, I'd like to acknowledge our funding sources, Minnesota Department of Agriculture, Forever Green, the USDA NEPA Kernza Cap, and FFAR. With that, I will take questions.
Great, thank you, Prabin. I did see one question come in the chat, so you could start with that one.
Yeah, I saw Peter's comment about, I'd be really interested to hear more about the implications of perennial wheat for the Kernza program, particularly on the commercialization side. At least as a breeder, like it's so far out, I haven't even thought about it. Really, I'm at the moment, I'm mainly focused on developing the population, making sure we have a really good population in place, and collaborating with other scientists, primarily the Land Institute, in developing a strategy on how we can lead perennial wheat, hopefully along the same path of Kernza, to make sure that it sees some success, but haven't really thought about commercialization, marketing. But that said, I did mention that I want to work with scientists in the food science department and the likes of Perennial Pantry, if there's enough grain, to see what can be done with the grain, because ultimately the market, if there's no demand, then the crop just is not, it's not going to be profitable. So, but that's, I'd say, a little longer term plan, unless the Forever Green leadership wants to chime in, or if there's Colin in the call, what he thinks about it.
Can you hear me?
Yes, Colin.
Yeah, Prabin. Well, yeah, I've been tracking the development of perennial wheat for sure. I think you're accurate, at least in my understanding, in terms of where it's at in its development. But I would say longer term, you know, if I understand the developments around perennial rice correctly, if you have a yield comparable analog that's perennial to an annual grain, like, isn't perennial rice being sold as rice? You know, like, it's a much more scalable pathway. But I don't, I'd say, have a grip enough on the state of perennial wheat to know if or when that would be possible. It's a longer term endeavor that I'm excited about as well. A longer term.
Hey, Colin, this is Lee.
Hi, Lee.
One of the things I'd say about perennial wheat is that trying to get it to slot into current wheat markets is going to be a big lift and probably a really long-term project. The perennial rice only has a small integration from the perennial to make it perennial. The perennial wheat has many chromosomes from the perennial in it, so it's you know, close to 50% of its genomic contribution is from a different species. So it's going to have very different traits than traditional wheat is going to. So I've, you know, I led the perennial wheat program here at the Land Institute for about 9 years, and I was always struggling with the idea that we're calling it perennial wheat because that implies that it's kind of the same species, and it's not the same species. It's a different species, and it's going to be quite qualitatively different. And so, yeah, I've always wondered if we need to reframe this as, you know, something different than wheat. It's not just wheat that's a perennial, because if that's the bar, it's going to be too high to make for a long time. I've eaten it, tried to make bread out of it. It's in some ways more challenging to work with, I think, in making products than Kernza is. Surprisingly, I was very surprised by that, but it has some interesting interactions between the perennial and annual genomes. But the seed size is much bigger, the yield is higher. But they're really going to be different plants. They're going to fit into different places. They're going to fit different places in cropping systems. They're going to be quite, you know, I'm talking about Kernza and perennial wheat are going to be quite different things. So, yeah.
Well, Lee, that's very helpful to hear. And it sounds like on the farming end might be perhaps more straightforward for producers, but as a grain will still be a differentiated market and uses. So
I think so. I mean, Stephen Jones's idea was that it was going to be just like wheat, that you could plant it on the sloping parts of your farm and harvest it all together to combine. That really proved to be way too high of a bar to be feasible to have something you can sell as a soft white wheat. So, but you know, maybe with genome editing, we'll fix it in no time.
I have a question as well for the perennial. So will you do the hybridization between the perennial with the spring wheat? You got seeds from those. Have you analyzed the perennial gene? How will the population segregate in that number of years if they more than 2 years?
I have not done that, but I believe Shuwen is doing that. I had a brief chat, and like Lee said, there's a lot of genome that's coming from, well, you know, a good share between both. And I asked him, hey, is it hundreds of genes? Is it 10 genes? And I don't think there's a definite answer yet, but the hope is it's lesser genes, so it's easy to kind of see if you know, markers or other ways you can identify these perennials. But
Yeah, my interest in the alpha breeding, we alfalfa after 2 or 3 years, their yield began to decrease. And
It-
if we can map the perennial gene increase from whatever number of genes by gene editing, increase the number of years of efficiency, that is really good news for farmers, but bad news for companies. So,
Yeah, no, I had the exact question with Shuwen, and again, to reiterate, he's trying to identify that, but the perenniality itself seems to hold quite well in Kansas, and we've seen it hold well for 3 years. Next summer would be 4th year in Jake's plots, and looks like that should be okay at least for 4 years if next spring looks fine. But beyond that, yeah, I don't know what exactly how many numbers and what kind of control they have.
is there a specific population designed to map the perennial gene?
I think so. I think Shuwen he's the creator of the germplasm that we have, and he has different segregating materials that he's looking at.
Okay, so my second question for the Spark products, they're really great products, and you take images from the root, then what if I understand you correctly, you will study the correlation between the underground and the above ground. And do you, maybe the roots and shoots is like an egg and chicken problem. Do you have a specific breeding goal to specifically breed a special root type to increase the above ground section, or you just study the correlation? Then what is your follow-up once you understand the correlation?
The long or the big picture idea is, at least my personal hope is that the root information can be incorporated into genomic prediction models. We rely on genomic prediction almost entirely when making selections for the next cycle, and say there is good correlation between some root morphology and seed size. I mean, I don't know if it's going to happen, just throwing an example. Then I would like to be able to use that in predicting root performance in upcoming progeny and the parents and select those. Now, is that possible? We will find out, I suppose, in the next few months. But as of now, we really do not know what we're going to find. I have a good guess that we'll have good diversity in terms of these root traits and the differences across genotypes. How will they perform in genomic prediction model? How will they correlate with above ground traits? And we don't know. And if they do correlate well, is that the correlation versus causation? I mean, we have to study that a bit deeper as well to see if it's indeed a true relationship. But ultimately, I'd like to be able to use them in my genomic selection models to make selections.
Yeah, I have a full root system architecture breeding program right now is in 5th selection cycle. So it's about 25 years of breeding for the root system architecture. And I'm really interested to use the same tubes as Dick Younger installed, and explore collaboration opportunities with you. So.
Yeah, we should definitely talk afterwards. I think we can definitely collaborate and see exchange methods and see what works the best. Yeah. There's a couple of questions in the chat I'd like to go through. Carmen Fernholz has any evidence relating to planting date on any of these or other perennials later to be able to follow corn or soybeans? Carmen, on the new crops, besides perennial rye, which I think is similar to winter rye, we don't know yet. I want to work with Jake, and Jake is also on board and running some of these tests on these new crops. Hopefully, and not too distant in the future, we'll have generated some like observations and data on this, and we can definitely share. Jeff Berg says, similar question to Carmen, what are these perennials? Winter, spring, facultative, fall seeded, vernalization needed, etc. From what I know, Jeff, they are all perennials and they all need vernalization. Kernza, we give in the growth chamber 6 weeks to 8 weeks of fertilization treatment. Same with perennial rye. Winter wheat, we don't know. I'm going to treat them similar to winter wheat, which is I think 6 weeks, 4 Celsius, and the winter type lights. I think it's a safe bet. Fall seeded, yes. Probably can also do spring planting. Like again, there's several agronomic studies where there's been tests on a spring planted kernza, so it should be possible. We just, yeah, a lot of things we don't know yet besides on kernza. So hoping we can do some of these experiments to answer some of these questions.
That's an interesting idea about getting your, if the perennial cereal rye can go in extra late, like annual cereal rye can, that would just simplify the rotational question going into it. So that's be interesting to throw some seed out if you have any extra lying around it, like November 1st. see what happens.
Oh, I got bags and bags, but it's good thought. David Marks has question: do you select for taste or nutritional traits? Not yet. Given in current,
that was just one thing. My thought was just simply that for the native populations where these plants arose, I would imagine the folks that selected plants know whether things taste good or bad and that kind of thing as part of their selection process in a native sort of a sense. And just wondering, yeah.
No, I've wondered that too. And in wheatgrass, we do mostly, space plants. In perennial wheat, rye, I think a latter generation, but rows-based evaluation will work just fine. On those, it may be easier to generate this data, but there's so many other traits that need bettering and fixing first that would probably come much after in terms of selecting based on taste. Now, that said, I would of course love to have data on space plants and then select the ones that promise better taste or better beer making ability and so on. But I think the resources need to probably be focused on some more pressing needs. But that would be one day, hopefully we will get there, because that would be an amazing data set to have and make selections based on that.
Okay. Well, thanks for a great talk. You've really been very informative with all the variety of things you're doing. It's very impressive.
Thanks, David. Excited to see what the mutants look like.
One issue that you didn't go into is this whole issue of speeding up the number of life cycle of breeding cycles in a period of time. And that debate goes on. The Land Institute is trying to really push the number of cycles within a period of time. And you tend to take a slightly different approach. I'd like, in terms of the speed of each of the cycles, and I've heard Lee talk about the fact that if you're running 2 cycles per year, it's going to take 16 years to get the yields up close to spring wheat, and you're running 1, so that's 32 years. What is your thoughts about how to speed up those breeding cycles?
The answer comes down to mainly, I think, resources and how much of that can be used where. I think they're doing 2 cycles a year starting this year, and the most of the work still relies on genomic prediction, looking at or taking data from the previous few cycles and making selections on the next 2 cycles basically. And the majority of the work, because you're selecting on seedlings, you don't necessarily have to wait for field data as long as there's good predictions coming from older cycles, so to speak, and apply that. But to genotype a plant is $10 to $12, and if you are selecting, right now I do about 3,000 to 4,000, so that's, let's say $35,000 to $40,000 a cycle. If you do 2 a year, that's easily in the realm of 90K to 100K with supplies and everything. And you know, I don't think we are there yet in terms of how much resources we can burn, especially financially, to get that up and going. But if that was not limiting, I think 2 would be achievable. We have just shifted to 1 cycle a year from last fall. I'm hoping to maintain that at least for a couple of years before.
To me, it comes down to success or failure, right? In terms of investment that could create a success in 16 years versus a failure in 16 years, right? So there's a limited period of time that we have to actually put a functional yielding crop on the landscape. So I was, you know, I was just interested that you didn't mention that issue today in terms of your thoughts, whether there's other ways of increasing or decreasing the length of these cycles. That was the question, I guess, was a statement.
Jeff, you think you can double our funding again? Peter, can you make that happen?
One other issue, Lee isn't on the line anymore, but you guys all know about, you know, high by high crosses. And when I last looked at the breeding material coming out of Minnesota, the breeding material coming out of the Land Institute, they look like they're separate. Have you guys ever thought about making high by high crosses across that germplasm?
We haven't done it yet, but we have definitely talked about that. And there's some challenges in shipping space plants that you've seen with your own eyes perform the best versus shipping grain that, you know, again, each seed is genotypically different, so it'll behave differently. There's some challenges in that, but we have, and I think it'll happen probably sooner than later. Just need to maybe drive from here to Kansas and back, and include Utah in the mix as well to get some of these good looking lines and put them in the greenhouse together.
Cool.
More questions for Prabin? All right, great. Thank you so much. All right, see everybody next week.